welcome back to another video lecture, this one on chapter 27. We're going to be looking at how we get energy from food. Now in the next chapter after this, we'll look at specifically how carbohydrates get converted, but this is more of a general energy from food, sort of big picture type stuff. Okay, so metabolism has a couple of directions. So metabolism includes building as well as breaking down. So catabolism is the breakdown process. Anabolism is the building process. And so anabolism is what creates glycogen. It's what creates proteins. It's what creates nucleic acids, DNA replication, all that stuff. Catabolism is breaking down proteins, fats, sugars for energy. You can kind of see that summarized here. So catabolism will take any of those things that we've previously looked at and ultimately break them down into small pieces. Proteins, that would be amino acids, polysaccharides into monosaccharides and triglycerides, into fatty acids and glycerol. And then those small molecules will either be used in creating new, bigger substances or will excrete them as wastes. And anabolism is just the opposite of that. Some of those nutrients that we get from our food will go towards the building blocks for our molecules. And this is sort of a, 6-step sort of summary of metabolism. In A, we have different carbons coming into the citric acid cycle. We'll go over the citric acid cycle in detail in this chapter. So various carbons coming in. Now, B is the citric acid cycle itself. So A is the carbons in various forms. B, citric acid cycle. C is those carbon dioxides. So those carbons that we ingest, we're ultimately going to oxidize and breathe out as CO2. D is the production of these reduced coenzymes, NADH and FADH2. E is going to be those enzymes transferring electrons and ultimately aiding in the proton transfer across a membrane. This is going to build up a gradient of protons that will drive the ATP synthesis, so making the high energy molecules. And then of course F is the making of those high energy molecules. ATPase is going to do that. And we're going to look at some of the details of this reaction, or of these processes. So first we need to kind of look at what a cell looks like and where this is happening in these cells. So just a recap of what cells can include. Our cells are a little more complicated than bacterial cells, which don't have any membrane enclosed organelles, little divisions within the cell. We do. We have a nucleus where all our DNA is kept. We have lysosomes, which are important for breaking down not just food, but also foreign invaders. So these are kind of like the disintegrators of the cell. What is the word I'm looking for? Like the garbage disposals, kind of. Golgi bodies are for packaging proteins and other things made within cells for transport to other parts of the body. And mitochondria are the site of ATP synthesis. These are organisms that are very similar in their DNA to bacteria that a long time ago became incorporated into a bigger cell and kind of started making energy for it in exchange for protection within the cell. And so these mitochondria are where the ATP synthesis happens. So this is an animal cell, and you can see some of the differences in those different organelles. So again, mitochondria are like bacteria. Nucleus, Golgi, site of protein synthesis here on the ribosomes. Ribosomes are these little circles right on the endoplasmic reticulum. Anyways, so here's the mitochondria, a little closer look at it. Now the mitochondria has a few features. Now it has the cristae, which are these folds, and then it has an inner matrix, and this is formed from two membranes. So the outer membrane sort of encases the whole mitochondria. The inner membrane is very folded and makes what's called the matrix. All these little folds and cavities. I think the cristae refers to the same, so different parts of the same folding structure. What's going to end up happening is that this intermembrane space in between the outer and inner membranes is going to be the site of our proton gradient, our pH gradient, where hydrogen ions are going to get pumped to be later used, kind of like we would use water in a dam to drive a turbine to make energy. These hydrogens are going to be driven sort of by osmotic pressure to kind of make a protein synthesize ATP. But we're going to have to build up that proton gradient, and that's where a lot of these other steps come in. So there's a few parts of the citric acid cycle, skipping a few of the early reactions. The main ones are the citric acid cycle, which can also be more modernly called the tricarboxylic acid cycle, and in the old days it was called the Krebs cycle. So tricarboxylic acid and citric acid both describe the components or intermediate molecules, so their names kind of make sense. The Krebs naming comes from the person who sort of figured out all of this stuff. And then there's the electron transport chain. This is where phosphorylation happens. And phosphorylation actually happens in two places. It happens during glycolysis, which we're not looking at here, which happens at the individual substrate of an enzyme. So we call it substrate level phosphorylation. It doesn't need oxygen for that. Oxidative phosphorylation is what happens in higher organisms in the presence of oxygen, and we'll look at why oxygen is so useful at kind of being the ultimate receiver of the electrons in this electron transport chain in later slides. There's some important molecules in this whole process. One of those is ATP. Not just ATP, but the intermediates that lead to ATP by additional phosphates. We're going to be trying to synthesize ATP in this whole process, storing that energy from the breakdown of food items. We're going to need some electron carriers, and two of them are NAD plus and FAD. Now these are going to carry electrons to the electron transport chain. They get reduced and then oxidized, reduced and then oxidized in an ongoing cycle that allows this whole process to keep going. And then we have coenzyme A. Coenzyme A is important for transferring acetyl groups, which are essentially two carbon groups. And let's see, what else? Okay. Oh, often it's abbreviated as coenzyme A with the SH shown here. This is an important functional group in the molecule. It's the site of where the connection to the two carbon acetyl groups happens, and so it's often shown with the SH. So here's ATP. We've seen this before. We saw this in the last chapter or two. What's great about ATP is that when we hydrolyze it to form ADP and then AMP, it liberates some energy. And so this is just kind of showing us how much energy we get per mole of essentially phosphate hydrolyzed. And this can happen twice. We can go ADP into AMP and get another 7.3. And so we want this molecule around. We can couple this molecule's hydrolysis to other reactions that normally don't have enough energy to happen, and that'll pay the cost. It essentially lets them happen. And one of the key reactions early on in glycolysis, which is something we're not looking at here, is that we get some ATPs already just from breaking down sugar into some of the molecules that can enter the citric acid cycle. So we're already getting energy just at that level. Citric acid cycle is going to produce a lot more ATP, and we're going to see how that gets done. But ATP is the whole purpose of breaking down these molecules. So here's NAD and NADH. I think at the moment we're seeing NAD. It's not reduced yet. It'll get reduced, it gains an extra hydrogen. Here we go. So here's NAD plus and NADH. So this this bond right here shifts over. Well, there's a couple of shiftings, it looks like. Lone pairs move somewhere, and we get another H added here. So that's the main point, is that it gains some electrons and a hydrogen, and it's going to take those electrons somewhere else. Here's FAD. This gets reduced into FADH2, and again, it'll also carry some electrons to the electron transport chain. So here we're just seeing where those hydrogens are going to go. And here's coenzyme A. And coenzyme A is carrying the acetyl group, which is this 2-carbon bit right here. And this is coenzyme A. So acetyl coenzyme A is another way that you'll see this molecule. Now, we've kind of skipped over the beginning reaction. I'm not sure quite why the book does it that way. So we can take a glucose, which is 6 carbons, and we take it through a series of 10 reactions that breaks it down into 2 3-carbon pyruvates. And this generates some ATP. Actually, 2 molecules of ATP for every glucose. And then we have, like I said, these 3-carbon pyruvates. Well, there's a reaction that'll liberate a carbon dioxide from each of these, so I guess times 2, and we're left with 2 of these acetyl coenzyme A's, where these are the remaining 2 carbons from that sugar. And then this goes into the citric acid cycle. So that's the job of acetyl coenzyme A, to transfer those remaining carbons into this next energy producing step. And this is acetyl coenzyme A. This is the SH way over here. That's the functional part. All right, now this is the very general sort of schematic of the citric acid cycle. So what you see here is acetyl coenzyme A coming in, dropping off that acetyl group. It's going to go through a series of 8 reactions. All the while, we're going to liberate 2 carbon dioxides. Now, these carbon dioxides that come out are not the same 2 carbons that just came in, but ultimately, those 2 will be breathed out in a subsequent cycle. We're also going to generate some electron transporting molecules. We're going to get 3 NADHs in this process. We're going to get an FADH2, and we're going to get some GTP, some substrate level phosphorylation of GDP into GTP. So we're going to make an energy molecule. And so this is going to happen twice for every glucose molecule that comes in, because we'll get 2 pyruvates and ultimately 2 acetyls. So I think we're going to take a closer look at some of those reactions now. So step 1 is actually getting that acetyl coenzyme A to let go of those acetyls. And so the molecule that it interacts with is oxaloacetate. Oxaloacetate will react with acetyl coenzyme A with the help of an enzyme to form citral-CoA. It'll dehydrate and form citrate, and we get back our coenzyme A. So coenzyme A gets reformed, ready to go do the job again, and now we've got citrate. So here in red are those two carbons that just came in. Citrate will get dehydrated into acetate. Acetate will get isomerized into isocitrate. It's interesting here that isocitrate is chiral, where these guys were not chiral. Isocitrate will only form one of the possible isomers, though. That's the beauty of enzymes, is that they control the reaction products very well. So we get isocitrate. Isocitrate dehydrogenase will then turn isocitrate into oxalosuccinate. Oxalosuccinate will then liberate one of those carbon dioxides. Now again, it's a different carbon dioxide, or a different carbon than the two that just came in, but ultimately the two that just came in will be part of the next oxaloacetate molecule that'll accept the next acetate, and so on and so forth. So let's see. Once we've got oxalosuccinate losing that carbon dioxide, it becomes alpha ketoglutarate. We can talk a little bit here about this enzyme, isocitrate dehydrogenase, since we know now a little bit more about enzymes. This is an allosteric enzyme that is inhibited by ATP and NADH. So the presence of ATP or NADH will cause the shape of this enzyme to change so that it stops doing this job. We don't need to make energy if we've got lots of it around. On the other hand, if we have lots of ADP, which you would only have if your ATP is getting used up, and lots of NAD+, which you would have if, well, I imagine if you have used up all your NADH. We haven't seen where NADH goes yet, so I didn't want to say anything, but if you already have, depleted your NADH, well then of course this is going to trigger this reaction and this whole process to go. Alpha ketoglutarate will now react with our dehydrogenase complex to get our first molecule of NADH formed. So this is an oxidation reduction reaction where alpha ketoglutarate is going to get oxidized, NAD plus is going to get reduced, and we end up with succinyl-CoA, and we lose that second carbon dioxide. So two things happen in this reaction. We have the reduction of NAD plus, and we also have the liberation of a carbon dioxide. All right. This is a multi-enzyme complex. Getting all this done. Again, inhibited by the presence of ATP, NADH, and succinyl-CoA. This is the direct product of this enzyme, or of this complex. So if this is already in high concentrations, then we don't need this enzyme to do this job. And so of course that would negatively regulate that. Succinyl-CoA then will phosphorylate a GDP to form GTP with the help of succinyl-CoA synthetase. We'll now have succinate, and we get back our acetyl-CoA again. Acetyl-CoA was part of the succinyl-CoA structure. And so now we have just generated the first substrate level ATP equivalent. GTP is a high energy molecule, just like ATP. Okay, a couple more reactions. Oxidation of succinate. So succinate gets oxidized here so that FAD can be reduced to FADH. We now have fumarate. And of course these are catalyzed by enzymes. Fumarate now gets hydrated into malate. So they add an H and an OH to this double bond, right? Reaction of an alkene. You can remember that. Oops. And the last step here, malate, is going to reduce NAD plus and itself get oxidized into oxaloacetate. So we've regenerated now the first molecule that reacted with our acetyl-CoA in the very beginning. And this process just repeats over and over and over. And so then the next two carbons that get breathed out will probably be if not the next round, the carbons that came in on this last sugar. And so this is how we generate all of those NADHs, the FADH2s, and the GTP. And that's shown here. For every turn of this wheel, for every acetyl group, 3 NADHs, an FADH2, and a GTP. And we breathe out 2 CO2s. So that's the big picture with carbon dioxide, with the citric acid cycle. It's controlled by a few feedback mechanisms. There are 3 enzymes that are under control. Citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex. We talked about these as we went through them. They all were inhibited essentially by the presence of ATP or NADH, some of them even by their own direct products. Catabolism from other sources, other food sources other than carbohydrates like proteins and fatty acids, can all join into this citric acid cycle at various points. So carbohydrates go through pyruvate, which we talked about, but didn't have a slide on it, but I was explaining that to you guys. Pyruvate becomes acetyl-CoA. Well, fatty acids can also go in at this point and be broken down into acetyl-CoA. Proteins can become pyruvate. They can become acetyl-CoA. The protein alanine, for example, is just a amino form of pyruvate. Proteins can become acetyl-CoA, and then they can also enter in as many of these precursors to amino acids. So when we make our own amino acids, we usually make them from these molecules. So different points where our molecules can come into the catabolism cycle. Now, let's talk about oxidative phosphorylation. So all of what we just looked at essentially happens in the cytosol. This happens floating around just in the liquid part of the cell. Now, all of what we're going to look at happens within the mitochondria, within the matrix of the mitochondria. So those, we're skipping over some of the details, but those electron carriers have to essentially get their electrons into the mitochondria, and they can't pass through the mitochondrial outer wall. So there's something called a malate shuttle, a fumarate malate shuttle, that basically transfers the electrons in, but then they go right back to NADH and FADH2. And so we can kind of pretend that they just were the same NADH and FADH2s that originally we started with, but they're not. What we're going to see here is that by pumping hydrogens across the membrane and by transferring electrons from these electron carriers to oxygen, we're going to form water and produce energy in the form of ATP. Now, organisms that don't have the ability to give the electrons to oxygen, they have to do something with those NADHs. And often what they do is they just use them to basically reduce pyruvate into something else. And so pyruvate will get reduced into lactate, or it'll get, it'll get oxidized. It'll get oxidized, no, I was right the first time. It'll get reduced into lactate in our muscles, or in like yeast, it'll get fermented into ethanol. And so I think we're going to go over glycolysis in the next chapter, but that's essentially what happens to the NADH when it doesn't have oxygen to take electrons to. So this is the general picture. So if you can imagine, we're saying the electron flow is this way. There's a few complexes. They do different jobs. Complex 1, we'll go through them, but complex 1 essentially takes the electrons from the NADH and uses that energy to pump hydrogens across the membrane into the intermembrane space, building up that concentration of hydrogen ions, of course lowering the pH. There's complex 2, which involves in taking the electrons from FADH. Now that itself doesn't pump the electrons or pump hydrogens across, but it does give electrons to coenzyme Q, which coenzyme Q is able to then pump a couple of electrons. I'm sorry, I keep saying electrons. Pump a couple of protons across the gradient when it gives its electrons to cytochrome C. And so there's a little cycle here in which that happens, and so we get some more hydrogens pumped across. And then we've got complex 3, which of course is holding those different cytochromes in these iron-sulfur compounds, iron-sulfur proteins. We've got this cyst C, cytochrome C, taking electrons to complex 4, where oxygen is, and giving these electrons and hydrogens to oxygen to form water. This also pumps another couple of hydrogens across the intermembrane space, again building up that pH gradient. And then this enzyme here, ATP synthase or ATPase, takes those hydrogens and like a water wheel uses that force to get this thing spinning. Excuse me. And when this thing spins, ADP and phosphate get combined into ATP. I have the hiccups. Okay. So all of that happens in the presence of oxygen. So let's go through it. So here's complex 1. Again, complex 1, large complex, a lot of subunits, flavoprotein, iron-sulfur protein clusters, coenzyme Q. This oxidizes the NADH to NAD+, which is so that we can take this back to the citric acid cycle, and it can keep doing this job. And then this moves those hydrogens across. We'll just talk about anything that's worth noting that I didn't already say. So this is what oxidizes the FAD back into FAD+, again, to go back into the citric acid cycle. Here we have complex 3, where we can see cytochrome 2 gets reduced by, it's not showing what it's getting reduced. The reducing agent was the coenzyme Q. Coenzyme QH2 gets turned back into coenzyme Q. So a handoff of electrons, essentially. And then after that handoff happens, we get some more hydrogens pumped through. Complex 4 is the one that ultimately gives electrons to oxygen. Now, there are organisms that do somewhat better than just reducing to lactate or to ethanol. They're called like sulfur fixing or nitrogen fixing bacteria. These have found a way to give electrons to nitrogen or to sulfur, which is getting more energy than they'd get by basically doing nothing with them. But oxygen has been kind of shown or can be seen to give the most energy. Okay, and so here again, hydrogens get pumped across. Water is formed as a waste product, and we see the total hydrogens that get pumped across are essentially 6 hydrogens pumped per NADH and 4 per FADH2 from the electrons brought by FADH2. All right, and so this is just basically telling us about that proton gradient, how it's going to kind of drive the synthesis of ATP, kind of like a dam with the water, the chemoosmotic pump. And so as it gets the different subunits of ATPase spinning, two different things start happening. We form ATP, and we also let hydrogens through. All right, so replenish our coenzymes to go back into the citric acid cycle, and we made ATP. So overall, how many ATP do we get? Every NADH gives us 3 ATP. Every FADH2 gives us 2 ATP. And I think we're going to talk about what we got from the entire citric acid cycle. So for each 2-carbon acetyl groups that go into the citric acid cycle, so 2, and it happens 2 times for every one sugar molecule, every glucose or fructose or galactose. We'll get 3 ATP for each of the NADHs. There's 3 of those, so 9 altogether. We'll get 2 ATP for each of the FADHs. There's 1, so we'll get 2 of those. We get a GTP in the process of citric acid cycle. So every turn gives us 12, and there's 2 for every glucose. So that's 24. And then there's other parts that we're not looking at, glycolysis, which gives us a few more. So a lot of energy per sugar molecule. We'll see that you get even more energy for breaking down fats and proteins. The different ways that we can convert ATP, this stored energy, into other forms of energy our body uses. One of those ways is the electrical energy we use to send our brain signals, our neurotransmissions. This ATP energy is what pumps potassium across the membrane into high concentration. It's what pumps sodium across the membrane into high concentration, so that when we open up the floodgates, these two flow across along their concentration gradient and send that electrical signal that we need in our brains. So we convert ATP, stored energy, into electrical energy. We can convert it into mechanical energy. I'll show you a diagram here in a second of how muscles contract, but essentially ATP drives that back and forth between the contraction and then the relaxation of the two proteins, the actin and myosin, in your muscle tissues. And that's ATP-driven mechanical energy. Your body heat is maintained by the hydrolysis of ATP, right? ATP to ADP gives off enough energy to keep you warm. You can actually stay warm on the coldest day of winter drinking an ice-cold soda, as long as it's not diet, because the sugar in the soda, as it's getting hydrolyzed and as it's being oxidized into carbon dioxide, as it's going through glycolysis and the citric acid cycle, is generating enough energy, just even right there, without oxidative phosphorylation, just in creating the ATPs and in the process of being broken down, keeps you warm. That's how much energy we get. And that's heat energy. Here is that muscle tissue, so actin and myosin. When they contract, they basically pull towards each other. Those are arrows, sorry. And then the fiber contracts, right? So the length shortens. And then we have them, we need them to relax, and then they go back to disassociating from each other. I think there's a neat video online that shows the little proteins that kind of walk along each other as they, contract. It's pretty neat. All right, so I guess that's the end of the chapter. All right, I think that marks the end of the material for the next test, but we do have a few more chapters coming. We'll be doing 28, 29, and 31 if there's time. So, all right, guys, see you in the next video.